Autism spectrum disorder, ASD, is a neurodevelopmental condition characterized by distinct differences in brain function, particularly within networks responsible for social interaction, language, sensory integration, and executive functioning. Individuals with ASD often exhibit altered connectivity between various brain regions, manifesting as both hyperconnectivity, excessive local neural activity, and hypoconnectivity, or reduced coordination between distant areas, such as between the frontal and temporal lobes, as well as between the prefrontal cortex and the amygdala. Genetic studies have identified mutations and dysregulation in genes critical for synaptic plasticity, such as SHANK3, NRXN1, and NLGN3 in many individuals with autism. These genetic variations affect the strength and stability of synaptic connections, thereby influencing learning, adaptation, and behavior. Sensory processing abnormalities are common, with individuals showing either heightened sensitivity or diminished response to sensory stimuli, likely due to disruptions in the balance between excitatory and inhibitory signaling, particularly within the cortex and limbic system. Notably, atypical activity in mirror neuron systems is frequently observed in autistic individuals. These neurons play a crucial role in interpreting others' intentions and emotions, as well as in learning through imitation and observation. Such differences help explain common challenges with empathy, facial expression recognition, and social communication in ASD. Overall, the autistic brain processes information in a more localized and fragmented manner, struggling to integrate multisensory inputs and coordinate activity across large-scale neural networks. Of growing concern is the marked increase in diagnosed autism cases in recent years. This trend appears to be closely linked to rising levels of nanoplastic pollution in the environment, present in the air, water, and food supply, which raises significant public health alarms. Nanoplastics, upon entering neural tissue, induce chronic inflammation, disrupt synaptic connections, and trigger apoptosis, programmed cell death, of neurons. These effects accelerate brain aging, impair cognitive function, and exacerbate neurodegenerative conditions such as Alzheimer's and Parkinson's diseases. Additionally, exposure correlates with heightened risks of schizophrenia and other severe neuropsychiatric disorders. Alarmingly, individuals inhale approximately 68,000 nanoplastic particles daily, with concentrations notably high near open water bodies and forests, as well as in urban centers. Even more troubling is the finding that nanoplastics have been detected in 100% of placental and sperm samples analyzed post-mortem. This indicates that exposure begins before conception, potentially compromising fetal development and increasing the likelihood of genetic mutations, serious health issues, and neurodevelopmental disorders. The ramifications of such prenatal exposure are vast and deeply concerning. Beyond these impacts, nanoplastics interfere with the brain's electrical activity, disrupt normal dopamine and serotonin regulation, impair microglial function, the brain's immune surveillance cells, and intensify autoimmune responses. They also contribute to rising antibiotic resistance. Collectively, these factors lead to a progressive decline in cognitive capacity across the population, a trend that meta-analyses associate with increased environmental nanoplastic contamination. Mental faculties and memory gradually deteriorate, and average IQ scores have shown a consistent downward trajectory in parallel with this pollution. Research on the effects of nanoplastics in honeybee brains further illustrates the threat. Even at low concentrations, nanoplastics cause significant impairments in memory, learning, and navigation by damaging neurons, reducing synaptic activity, and increasing oxidative stress. While the honeybee brain is far simpler than that of humans, it similarly relies on precise neural network function and synaptic plasticity for adaptation and learning. Comparable neurophysiological disruptions have been documented in mammals, including humans. Accumulation of nanoplastics in the human brain leads to analogous cognitive deficits, memory impairments, and learning difficulties. The honeybee studies serve as a stark warning. If nanoplastics can disrupt neural processes in such small, sensitive organisms, their impact on the far more complex human brain is likely profound and far-reaching, especially given the ever-increasing levels of these particles in our environment and bodies.